one of the few things that is the government does that makes sense here. But all right. Well, we just started recording, so we are live. Nick is coming to us from where you at in the Bahamas?
I'm at Elbow K at Sea Spray Marina aboard
Okay.
the Risk and Reward. It's a 55 Rabovich.
So if he feels the boat go into reverse, he's gonna have to run out of the.
Dude, I'm like, hey, you know, something happens. I start hearing things. Pump starts going off. I gotta go.
Yeah. But yeah, thanks for making that happen and get with us. And then Miguel's coming from Florida. So thank you for making this happen, man. And I really appreciate it. This is a little different than any of the other ones we've done. And I'm excited because I really kind of enjoy all the science aspects and all this side of Billfish. We spend so much time chasing these things around the world, and a lot of us don't know that much about them. So it's cool. And what's interesting about what you do is one of the main things we're going to go over is teeth, which I'll be honest with you, I didn't really know that they had teeth. I mean, I kind of thought, but it wasn't something I thought about very often. So anyways, if you can give us a little rundown of kind of, who you are and what you do and all that good stuff.
Awesome. Thank you, Chris. Thank you for your invite to be here. This is my first time doing this. So it is really exciting. So my name is Miguel Montalvo. I am a graduate student at the Virginia Institute of Marine Science. My background is not in marine science. I was a marketing major from the University of South Florida. And then through different things, I got into grad school. I really wanted to do something sciencey at some point in my life. And this was just a great opportunity. And I started grad school in 2022. And now I'm studying billfishes. So it has been a good little bit more than 3 years of a deep dive into the biology of billfishes, learning something completely new. I got into fishes because I was working before that at the Smithsonian, and I had a friend that worked at the Division of Fishes, and she got me really into them, and it was just an amazing experience. And then an opportunity during COVID opened to apply for this program, and I'm here. And and that's what I've been doing. I'm studying the teeth, the fossils of billfishes too, and then I'm also looking at the evolution of rostral elongations among fishes, because they're just such a cool feature that billfishes have. It gives them their name. And there's so many other fishes that also have an elongated snout. And it's like, how did evolution mold this into what kind of environmental factors, what kind of things lead to them? So those are sort of my three things that I'm studying, my 3 chapters of my dissertation.
How long is your PhD program, Miguel?
So this at VIMS, it's kind of weird. You usually start as a master's student, and then you bypass into a PhD.
Okay, I got you.
So it makes it a little bit longer, but people usually graduate around like 5-6 years. There's some people that, due to what they're doing, they take a little bit longer. But it's good that the school has that sort of flexibility. Yeah. But so far what I found is that graduate school is great, but it keeps you busy all the time. Like, I've never been this busy. I was not this busy when I was working like professionally or anything. Like, this is the busiest time of my life. Yeah.
Right on. Well, there's a lot of stuff to unpack there. There's definitely some cool topics we can go over. So kind of got a lot to cram in here. So we'll start off first with with Billfish teeth. And then, anytime you want, you throw up pictures and all that. He's got some cool photos of Billfish and teeth and different things. But yeah, man, I'm gonna let you. I'm gonna kind of keep quiet here and let you kind of talk to us a bit, and then questions as you go. But I think Nick and I are both just interested to hear what you got and learn a bit about their teeth. So yeah.
Yeah, I agree.
Okay.
Go ahead.
So, my thing with teeth of billfishes is that people really don't know that they have teeth because they're minuscule. They're tiny, tiny, and they have so many of them. They don't only cover like their jaws and for many species the underside of their bill, except for swordfish that as an adult loses them. So a juvenile swordfish would have those teeth on the underside of the bill, but as an adult, it loses them. All the other billfishes, they have them on the underside. And there's some crazier ones, I've seen some sailfish with teeth that go like above the rostrum, and especially on the tip. We still are not certain what those teeth do. Billfishes do strike their prey to eat it. And it might be to increase damage to the prey. It might also be because of some weird hydrodynamic reason that we don't know. So that part of like the development of this fish putting in so much energy into putting teeth on the rostrum is just mind-boggling. So that's a little bit of something that needs more research, and it's not something that I am doing. What I am doing is seeing how from little tiny larval billfish, which they start off as millimeter in size, and then they grow the size of a big blue marlin. Can be what is the record, like 1,200 pounds or more to 1,500 pounds?
1805 for Henry Tree, right, Chris? I mean, it's not the official record because it was two people called it, but that 1805.
There was an 1,800 pounder caught in Wahoo by that Choi.
Holy moly!
Yeah, the record blue is here in Kona, Hawaii, 13.
Oh no way!
13 something or other, but yeah, they get very big, and there's been 1,600 pound fish caught. There's been giant ones caught over the world record, but IGFA world records, the 13.
Yeah. The Atlantic world record, I think, is Brazil at 1,400. I think
yeah,
in the 90s.
Yeah, 1,400 for Brazil.
Yeah.
Oh, wow.
The Pacific has the bigger non-IGFA catches. The Pacific has the bigger.
Okay. I have the biggest one that I have seen is like 800 pounds at the Mid-Atlantic here, and I thought it was humongous. And just to think how something that little grows into a humongous fish in really a very short amount of time, because they're super fast growers, is just crazy. And the big changes that go on their dentition from when they're super tiny to those adults are remarkable. So that's what I am studying about their teeth, is how they manage to change those teeth from when they were really tiny to fully grown adults. So here, let me show you a photo of a clear stain. So this is so there's two families of billfishes, Ichthyophoidae, which includes all of the billfishes except for swordfish. Swordfish is in its own family, which is Siphidae. And when they're really tiny, you cannot tell them apart except via genetics. So I have an pretty much unidentified Ichthyophoid billfish here, and this is the larva of one of them. So it could be a sailfish, it could be a round scale spearfish, and this specimen came from the Straits of Florida. It looks this way because I have prepared it using a technique called clearing and staining. And what this technique does is that the bone gets stained red and the cartilage gets stained blue. And then it becomes transparent because we put it on a digestive enzyme called trypsin. And the coolest thing is that this enzyme eats up all the muscle, all the stuff that's not transparent, and leaves a specimen like this, which just happens that the connective tissue that's left behind is the same refractive index as glycerin. So if you put it under glycerin, it looks completely transparent. If you put it under water, it looks a little bit whitish. But as you can see, this is 2 millimeters in length, or a little bit more. No, probably a little bit more. I think this might be like a 4 millimeter in length fish. You can see it has massive spines, and it has a ton of teeth
Look at those
in there, right? They're gnarly. They're gnarly for this size fish, right? Like
Yeah.
they're
Dude,
big.
Legit teeth.
I know,
Yeah.
right?
So at that size, at that age or whatever, are they actually using those teeth to feed
Yes.
on what they're eating?
Yep. And let me see if I can find another photo because they are using, definitely they're using them to feed on stuff. And one of the things that they feed on something, let me see if it's this one. Don't have a clearance thing. No, I'll not show that one. It's just not. But we often find them with other billfish in their stomachs.
Even
They're
oh
billfish.
wow, so they're eating
Yeah,
each other at that age.
even that tiny, they're eating
Wow.
each other. And let me see because I do have another photo of a one of these with
So
prey on.
that's something I kind of always wondered was, will they eat their own species? Like we'll find
Yeah,
fish. Sometimes guys will cut a big one open, and it'll have like a blue marlin will have like a striped marlin in its stomach, or like a spearfish or whatever. So I was curious if they will also eat their own kind. Like if a blue marlin will eat a blue marlin, or because apparently tunas don't do that, right?
oh really?
I don't know. I didn't hear that scientifically, obvious, but
Mhm.
I've just heard that they something to do with colored patterns or something, and how they would tell what is in their same school and what is not. But I guess they're different because they're schooling fish maybe. So I could imagine if you had a pile of yellowfin, they're probably not going to attack each other because they're in a school where.
No, but that's an interesting point because, like, I mean, we live bait back home. I've never caught a yellowfin live baiting like a small chibi. Like I've caught them live baiting a blackfin or a bonita, but I've never caught them live baiting it.
Right.
That's yellow. It's kind of
yeah. So I'm not sure if anybody out there knows that if that is actually fact or not. Please let us know. But I have heard that several times that tunas will only eat other species of tuna. So I was curious with the billfish if that's what they're doing, but
Yeah, they're eating for sure other billfishes, but I don't know if from their same species or from different species. Can they tell like the way that we cannot tell what species they are except via DNA? Like, can they tell it somehow? But yeah, they're remarkable, and they do eat a lot. I wanted to find a photo of one with stuff in its stomach, but I cannot seem to find it. But you usually find just really large prey for their size inside of them. And those teeth are probably a lot to blame on that they're eating something as big, right?
yeah.
And so this was one family that was Istiphoridae. And then this is a larval swordfish, which is the other family. And they also have big teeth, also equally prepared via clearing and staining.
Oh, cool.
And you can see that they have really neat teeth. And
I was not gonna say it, Chris. I didn't want to sound all Louisiana.
But it kind of
I was
does.
like, it looks like a gosh dog guardfish.
Yeah.
Garfish.
Like
I
a
mean,
garfish.
Yeah.
Yeah, yeah.
like, I didn't want
Uh,
to say it. I'm glad you did.
yeah, no, they're just remarkable, and I think swordfish, their their rostrum grows a lot faster than the other family during at least these sizes, because whenever I see them, they're a lot more pointy. They have a lot more a longer bone
Yeah.
in there.
Their build
And
definitely bigger.
no, sorry. What were you saying, Nick?
No, I was like, yeah, their build definitely gets bigger. I mean, quick. I mean,
Yeah, and then also the way that those builds are built as adults is very different. So
Yeah.
for
really,
they so swordfish, they're flat, sort of like a sword, and the other billfishes, they have more of a round cross
Right.
section. Um, so yeah, they're very different morphologically. The family that holds the marlins and the spearfish and sailfish, they're very different to what swordfish look like. But yeah, so those were the teeth when they were little. And let me now show you what billfish teeth look like if you zoom in as adults. So this is a blue marlin. Hold on, the photo's loading because it's a humongous photo. There you go. Yeah, I am a pixel. What is it? A pixel. I like to zoom in a lot, so I always save files on their highest resolution. So this is yeah,
whoa,
this is a zoomed in section of the lower jaw of a blue marlin. Um, oh no, sorry, of a swordfish. And you can see that they have all these teeth. It's interesting that there's some teeth that are bigger and like more stout on the side of the jaw, and they become a little bit thinner in the middle. So also part of my research is looking at this overall morphology of the teeth as like in different sample places, um, because it's just very interesting. Another cool thing that we've found or we have observed, is that there's little holes that you can see, and you cannot see it that well in this specimen, but there's little tiny teeth growing out of them. So they're actively replacing their teeth. And I do have another photo of that here. Yeah, let me share it. I'm just gonna leave this on. So this is from a white marlin, and you can see that there's fully grown teeth, fully developed teeth here, and then there's little ones coming out from these holes. So that's something that, to my to what I know, it has not been documented. So another part of that chapter is also seeing how that tooth replacement process works, if there's any sort of pattern to tooth replacement, and especially during ontogeny, during their development. Uh, see how that happens. So if you can see here, this grid, this little square is 1 millimeter in length. So you can see that they're really, really tiny teeth. Um, I have another slide here
oh, wow.
of
Those are
I
actual teeth.
yeah. So I dissected some teeth out of that same specimen on different sort of stages of development, and you can see that there's a big teeth that was being used, and some smaller stages, starting with this little tiny thing. If you can see, the tip is a different color, because for fishes that are not lobe fin fishes like coelacanths, they have this thing called the acridine cap, which is a different type of enamel that grows just on the tip of the tooth. So yeah, and you can see 1 millimeter, so super tiny. And that's why when you're grabbing a bill of a billfish or something, you have no gloves or anything, you can find yourself with little scratch marks, little cuts,
Yeah.
because of these tiny things.
So that's teeth then. The
Yeah.
like
Yeah.
we grab their bill and we all up and down the bill, all throughout their whole mouth, everywhere. That's all teeth. Those little same
Those are
teeth.
teeth. Yep,
Oh wow.
those are teeth.
So those are when we get marks on our lures when they bite it. Those are
Yeah.
we always call them bill marks, but they're teeth. They're teeth marks.
They are teeth. And so basic science. You start with sort of a question, right?
Yeah.
And you work through it, but you don't have an application in mind right off the bat. You're just like, I am interested in this, and I'm gonna explore more on it and see what happens. But through this work of finding these little teeth, which they have been documented a long time ago, but their development and everything, maybe you can think of some sort of bio-inspired material that uses these tiny sort of structures, because billfishes have those teeth there for some sort of reason. And maybe that reason is because they're just really good at grabbing slippery little tunas and mackerels and whatnot, right? So maybe some sort of bio-inspiration from those tiny teeth could be the next step on sort of like this basic research. So that gets me excited. I really like the basic research, don't get me wrong, but applications further down the line are always very interesting. And another thing that also could relate to fisheries management and all that is that juvenile stages of billfishes, which we really don't know much about them because they're difficult to get, they're difficult to find. Maybe we can tell something about their feeding behaviors from the tooth morphology. So that's another part of it. And I don't know if you probably have seen juvenile billfishes before, but I just think that they're the cutest things ever. And so here's a sailfish, a preserved sailfish.
Put some photos up. Yeah.
Yeah, let me see. So here's a juvie sailfish, so the 1 centimeter here. But look how cool that is, right?
Wow.
Like,
Yeah, there you go.
I mean, and this is nothing because like when they're alive, they're beautifully blue, right? And
Yeah.
like they just have staggering coloration. It's because we preserve them in ethanol and everything that they lose their color, but you can still see how remarkable they are.
Oh, you can see their
And
teeth.
you can see the teeth.
Dude.
Yeah, right. And from here, and I have not gone too deep into the juvenile section of my research, but you can see that there's tons of teeth in their lower jaw, and
Look
the
how far
upper
it's
jaw.
going down the bill. Yeah, that's what's
Yeah.
how far it's
And
actually traveling down this bill.
yeah, exactly. But they're more spaced out, right?
Mhm.
So, like at this point, the teeth that go in the bill have not filled out as much as what we see in the adults. So, when is that happening, and why does it happen, right? Like,
Mhm.
at what point during their juvenile stages they start using those rostral teeth, those teeth on their bills, and for what reason? So, that's
And you
that's
said there was teeth on the top of the bill too, on just
um,
sailfish or something, or
I think that there's other billfishes that have that. It's like for sailfish, the two specimens that I've seen, they have teeth on the top, but only on the tip of the bill.
okay.
But I was looking that there's some other species that have them more towards the sides, but so it varies, and who knows? Maybe that's a specimen to specimen sort of
Genetic.
variability.
Yeah.
but yeah, next time that you see that you go fishing and get a billfish or anything, and just look at their teeth, see where they are at. It'll be very interesting, right?
Yeah.
To look at that. So that's pretty much what I'm doing with my tooth billfish teeth chapter. And it's proving to be really fun. I am using different techniques. So you saw the clearance staining. I'm also doing micro CT scanning. So it lets me reconstruct a 3D model of the jaw, of the teeth, and just explore it a little bit deeper. There's some cool things that we have preliminarily found in the CT scan of one of these juveniles is that the teeth on the bottom jaw actually are not they're stuck to a what is called a bone of attachment, but that bone of attachment is not that strongly attached to the actual jawbone. And that's something very preliminary that I've seen on some of those CT reconstructions, but it's just very interesting. And then another technique that I'm also using is photogrammetry, which it lets you reconstruct a 3D model of whatever you're doing just using photos all around of it. So you can take several photos around a thing and you get a 3D model. And I am using that to just take really close up macro shots of the teeth to be able to categorize them better. And with that comes another whole amount of things. So like 3D printing to make models and all that. So it is. I truly enjoy working on that. It's just fascinating. Yeah.
Oh,
Yeah.
Sure.
that's really cool. Yeah, I mean that's definitely something I didn't think about. Now, we all know how rough their bills and face and all that is, but yeah, I'd never really kind of thought that that's actually teeth. So
I
that's
never
Yeah.
thought that was teeth ever. Thought that
and
was
it
teeth.
makes sense because when you think about it, like Nick, when we get lures that get all scraped up by them,
Yeah, we
it's
just
like
thought,
little tiny teeth marks, just like
yeah,
little.
and you to your point, Chris, like how it's not like a nick, it's like all the way across the head, which would make sense if he's grabbed, like it's getting too
Yeah.
much, not just like a smooth like clip. You know what I mean? Like that's
Yeah.
a good point.
And that, and I don't know if Miguel, if you guys have been able to, kind of go into these questions or not or whatever, but if you have ideas of what they're at their adult age or their, when we're fishing for them, right? What they're using, if the teeth are just to stun the fish as they whack it, or if it's used to kind of guide them in, like almost like a fork and knife, like to kind of guide it into their gullet there, and so they can take it down, or what, because they don't really chew them too much. I mean, I guess sails do it.
No.
For the most part, they kind of
Sales dog bone the heck out of them. I don't know if it's
Yeah.
definitely like feeding them like here, not like, but like here, these small Atlantic sails, like you have to like feed them.
Yeah, really give it, but they're not like chewing it up like we would.
No,
like they're not
it's like
chewing
they
it up,
hold
get it into
down.
tiny little pieces and swallow the pieces. They're kind of like smashing and arranging it and head 1st or however, and then they
Right.
down.
Yeah.
So
They're
have you guys?
basically dog boning it, Chris. Then they'll flip it.
Yeah,
Like
like a
you
pelican.
almost see the difference, and that's when you can set the hook kind of thing.
Yeah, kind of like a pelican.
Yeah, so the teeth in the bill, I think that's still a mystery. During the summer, that this last summer, I went to a course in Friday Harbor Labs up in Washington. It was a 5-week course, and you had to come up with a project to do during that course. And my question that I wanted to see is if those teeth were aiding somehow the fish on it's swimming, or maybe it's swiping, like hydrodynamically. Maybe it would help stabilize the bill or something like that. So I made some models, put them in a hydrodynamic flume, which is just like this thing that moves water along, and then you have sensors to see how much the model moves or what forces are imparted on it. And it was really cool. But sadly, the thing broke before I could like finish all the analysis, and the data that I had collected before was not too good. There were problems with the sensor, with the way that I was mounting it. So it was a course. I had to learn something from it. Now I know what not to do. But it is something that I would like to do in the future, is revisit that part of the project and actually see like, hey, what if teeth are just acting as some sort of like stabilizing mechanism for when you're swiping really fast, or who knows what other reason. So but yeah, that's still very much in like, what do they use them for, or is it just like to impart damage to the prey?
Yeah.
Because, and if you think about it, in swordfish, that they don't have teeth as adults, but they have really sharp edges. They swipe through a fish, and like you see slash marks on their stomach contents, of swordfish. So maybe that's the reason. And for sure, teeth help like position prey, right? Just not only because they're holding the prey inside like a certain way and then move it and don't let it slip out. But you also have to think that inside the mouth, so the teeth on the jaws, the bill and the upper jaw sort of are only a fraction of the entire amount of teeth on the mouth of a billfish. So if you open the mouth, they still have very raspy feeling stuff on the top of the mouth, on the mouth roof, on the mouth bottom, like on their gills. They also have a ton of teeth, and these are all sort of like homologous structures to these. So like, yeah, for sure, they're definitely there to grab and help move down the gullet of the fish, the prey item.
Yeah, that's so cool.
Yeah, they're really cool animals.
Yeah, there's so many elements to these things that how much they've evolved and just to become the predators that they are. It's really impressive. You were talking a bit about, you've done some research on their bills and the growth and all that kind of stuff. I would be interested to hear a bit about that.
So on the growth of their the bone components of their bill and everything, I am not doing that research. A colleague of mine, who's also a close friend or has worked with the Billfish Foundation before, Laura Habiger, she's working on that development. So she's doing some really cool work on how those bones develop, like what bones get elongated first when a billfish is growing and all that. So she's doing that work, and it's fascinating because like, what the heck, we have this thing, and like the bill is actually the premaxilla, which are these paired bones that just join up there, right? And those are the ones that have the teeth. But in the base, for like the swordfish and the other family, they are very different. So swordfish don't have a pair of bones called the prenasals, which the other family does have them. Why? Where in the evolutionary history it gets lost or it gets gained by the other ones, we don't know. It's just interesting. Also, in the lower jaw, another feature of the istiophoriid billfish, so sailfish and all of that, is that they have a tiny little bone on the tip of the lower jaw, which is called the predentary bone, and swordfish don't have it. So another mystery, when did they evolve it, or when did swordfish lose it? And so all of that is just like, it's a mystery upon mystery on these species of animals that are very charismatic, and people have looked at a lot, So it's really cool work. Yeah.
Yeah, that is really, really very interesting. Gosh, there's so many elements to it.
Got so many guesses now. He's talking about like, oh, I think this is why that. I get so many guesses, but obviously nothing concrete. It's so interesting.
Yeah, no, and like what you do is that you make those questions, and then you're like, okay, I gotta go and test it. Like
Yeah.
see what makes the most sense, what sort of agrees with like the evolutionary history of other fishes, and yeah, it's unending work of trying to like decipher evolution and how all this, like our current life forms are what they are. So yeah, it's really interesting. And if you want, now that we're talking about more evolution, I can move into the fossil part. Okay. So yeah, so billfishes are really interesting. We have some fossils. I am not an expert. I wouldn't be able to tell you compared to other fishes, sort of like fossil record, if we have more or less, because I really don't know. But their fossil history starts after or what we can tell starts after the impact of the meteor that non-avian dinosaurs got wiped out on.
Right.
And so we find the first sort of like what we think billfish fossils in this era called the Paleocene, and that's only a few million years after the impact of the asteroid. And these things are, of course, not nearly as big, or what we have found are not nearly as big as the big adult billfishes that we see. They have elongated snouts, and they have features that are very much like what we see on billfishes nowadays. And one of them, which is probably my favorite feature of a billfish, and let me see, where do I have a photo of that, is their backbone. So let me hold on one second. I did not think about opening this before. But their vertebrae are just really, really cool. And if you have the opportunity to pick a little bit more on like a filleted billfish, you'll see that in the vertebrae they have these bony projections that are really flat. And let me see if I can find that a photo of one here. I think this would be here. Let's see if it opens. These are just really cool. It's opening. Let me see. Oh, not this one. But in those fossil billfishes, you can see some of those features, that those little vertebrae. And I think I'm getting close. Oh yeah, here it is. So let me share screen again. So this is the backbone, and yeah, this is the way. So and this is of a white marlin. It is pointing forwards to the right, and these are those bony projections that, as far as I can tell, scientists have called them lamellae, and they're really cool. And we see them in fossil billfishes too, as preserved in there. So, and this is not the only feature, but there's sort of things that sort of tell us that those are fossil billfishes, but that's still a little bit up in the air. Another cool thing is that they have these sort of projections that make vertebra sort of like uh, fit into the next one, and they make a very very rigid sort of like vertebral column. So that like if I this one, it will take me a while to pull apart one vertebra from the other one.
Mhm.
I know that some people have studied if that's just to make them better at swimming by having a stiffer um, vertebral column or not, but I think that the results of that were like maybe maybe not. But in fossil billfishes, so here's an illustration of one of them. So this is a specimen of this family that I think that you're gonna like the name. It's called Hemingwayidae, which after Hemingway, right?
Yeah.
And this one is from these are the oldest sort of billfishes that we think. And this one's from the Eocene, from the Paleocene of Turkmenistan, and you can see that they do have those little like leaf looking um,
Wow.
flat spots on their vertebrae, and they of course have an um, elongated snout, and they have two dorsal fins and two anal fins, which is something that you find in current billfishes. And the tail, it also has this sort of thing that's interesting, which is called hyperstagy, if I'm not wrong, is that the fin rays, so the little elements that make up the fins, actually go in like in the caudal fin, they go on top of another piece of bone, which is several fused bones, which is the hyperals, and they go on top of them. And you find that on these fishes as well, which is another feature that sort of relates them to our modern billfishes. The specimens that I'm working on right now are not as old as Hemingway, but old. They're also old. So they are the next sort of era, which is the Eocene.
Mhm.
And these specimens are really cool because they were found on an expedition to Pakistan to look for early whales, and they're stored at the University of Michigan. And what like some they went to this expedition. I think it was back in the 70s, and they brought early whales and also a bunch of fishes, and they just put them in a drawer, and then or in a few drawers. And then my advisor and another committee member, who's Matt Friedman from the University of Michigan, they applied to a grant to look at those fossils and see sort of like what was the fauna of that era of that area in that time.
Mhm.
And amongst those fossils are the ones that I'm studying, which are they belong to another family. It's not as fun as Hemingwayidae. It's called Paleorhynchidae. And these things really don't look like much. And let me find some photos, because I did take a ton of photos the last time that I was at the University of Michigan looking at them. They're not the best preserved specimens, uh, but they're very interesting for sure. And just oh, this is gonna take a while to open. So I might talking about resolution. The photo that I took, it's a compound, so it's like maybe like 30 photos put together. So the file is like 2 gigabytes.
Oh wow.
Yeah, so
Oh my
it's opening right now. Um, yeah, and I don't store it in my computer. I have uh, a network attached storage for that, so it takes a second for them to open. Hold on.
They found that like it's wild. Never would have thought about it like that
but you're gonna be surprised at how much they don't look like anything until you,
Yeah.
yeah, they just look like rocks, which they are. They're just
Right.
rocks with with some fossil um, material on them. Um, and the fossil material is already rock. But so here it is. So this is one of my specimens from there, and yeah, pretty much a rock, right? uh, and you cannot tell much about it. I've annotated this one a little bit. Yeah, I
Oh,
can
cool.
the
Okay.
notes out of it, but you can sort of see
Yeah. Let's see
the
it.
general
Yeah.
look of the
Yeah.
fish, right?
Yeah.
So the eye
Big eye.
here, and then the bill and the lower jaw coming out here. You also have some dorsal fin rays that are really long, and you can also see the vertebrae here. This one does not have those flattened things preserved, or not that I can find. But we're gonna CT scan this one, and maybe there's some under the rocky matrix.
Yeah.
But yeah, and then I did take another close up of this under better light, so you can see it.
Right.
And yeah, you can see some of the features of this thing. Billfish eyes are really cool. Not only because I mean billfishes are visual predators and they have humongous eyes, but also their eyes are bony. They have this thing called the sclerotic bones, and they just ossify. Why do they ossify? My guess is that probably because they're swimming really fast and they don't want the water pressure or anything to modify the shape of the eye, perhaps. They're not the only fishes that have it. I think tunas have it too. Um, and they're actually not the only animals that have it, because owls and eagles, they also have eyes that have bones in them. So you can see part of that, the sclerotic bones here, which are interesting. And this specimen I really like because you can see some teeth on
Yeah.
them. I don't know if you can sort of see the little
Yeah. Yeah.
teeth in,
Absolutely.
and it is from what I've from my dive into fossil billfish literature. There's very few specimens with teeth um, on them. So this is another thing. Like teeth just keep creeping out into different like facets of my research,
Yeah.
and this is something that I'm really excited to look at, fossil billfish teeth as well. Maybe they hold some interesting things about, I don't know, their feeding behavior, the relationship to other fishes, or not. So yeah,
Even if
just
not, it'd be great to see how they evolved across all the years that you have fossils for, just to see how much they evolved across time.
yeah,
Yeah.
how they change through time. There's some running hypotheses about some features of old billfishes. One of them being that they, unlike modern billfishes, that they had bills that were the same length as lower jaws, and you can see that in this specimen and some other ones that I have, that both things are the same. Both upper and lower jaw are the same length, which is unlike what we see in billfishes today.
Yeah.
Um, so, and there's of course more modern specimens. Some through like the Miocene, Pliocene, which some of them are from modern billfishes. Interestingly, the like we have very few fossils of swordfish or fishes that are more like swordfish for some reason. But if you think about it, like why swordfish also grow to be really big fishes, right? Like the marlins and all of them. But for some reason, we don't get as many fossils. Maybe it's because they don't live in places where they would fossilize, or their bodies would fossilize better. But it is interesting that they don't. There's actually, like, I think the coolest billfish fossil, just because of where it was found, is a vertebra from what is it, Seymour Island in Antarctica. Someone found a vertebra,
Wait,
a
what?
fossil vertebra of a billfish
Antarctica.
in Antarctica.
That's
Yeah,
crazy.
of course,
Yeah,
it was from a time when Antarctica was a lot more tropical, right?
yeah, for sure.
But it is, it is interesting. And like, I don't remember if I was able to see the specimen or not, because someone told me like maybe it's iffy, maybe it was not a billfish vertebra, but who knows? Like, it's really cool. Anyways,
That's
yeah,
interesting. How
yeah.
the fossils and stuff, and that the picture that you showed of the old billfish, how big did they get, or how big did you guys, see them
So
too?
the specimens that I'm working with are probably like this size. They're not longer than 10
Yeah,
inches.
but they must have
And
grown pretty large back then too, or I mean,
so I think that early billfish fossils are not that big. We haven't found anything that are that big. I was reading on an earlier specimens, or later specimens. There are some that are meter in length, um, or so, but nothing like the monsters we see today. Like that, and this is back in their history. If you go like to um to the later geologic time, you start seeing fossils of billfishes like the ones we have right now.
yeah, right.
So, and there's some really cool ones, including vertebrae from South Carolina, I think from Virginia too. So yeah, and there's a lot of work that has been done on the fossil history of those billfishes, the marlins and spearfishes and sailfish, and that has happened in the past like few years. There's another colleague who has described several new species of those fossils from Panama, and other ones from Italy, and it's just like it's really cool work.
Right,
Yeah.
right. So theoretically, something must have happened, like in their evolutionary track, that allowed them to grow bigger, or enabled them to be more efficient, and you know
Yeah.
create become bigger. So that's kind of interesting.
Yeah, that's one of the hypotheses. Also, it is very interesting. My the committee member that I was telling you from, University of Michigan, and some colleagues of his, they are really interested about the time right before the impact of the asteroid and right after. How did the fauna, the like different faunae of or the different species change through that time, right? Because there's a lot of things going extinct.
Right.
And on one of their studies, they show that big pelagic predators, like some that were, what is it, pachycormiformes? They're big, fast swimming pelagic fishes, and how they disappear after the impact of the asteroid, and afterwards, how fishes like tunas, billfishes, and all that, they start filling up the void left by them, like getting like become the fast pelagic predators. So that's a really interesting sort of transition, from the sort of like the old lineage that got extinct to a new lineage that sort of took over that sort of diversity. So yeah, it is really cool that those studies that look into those species compositions through time are really interesting.
Hmm.
Yeah.
Yeah, that is cool.
Yeah.
Uh, it's interesting to see that picture you put up of like the early billfish and like how much they've changed.
Yeah. In so the oldest one that illustration that I showed you, the Hemingwayidi,
Yeah,
so family Hemingwayidi, and the genus is Hemingwaya, and it's there's only one specimen, I think, of it. And I showed you that it had two dorsal fins, two anal fins.
right,
In the fishes that I'm studying, they only have one. They have one dorsal fin and one anal fin.
right.
That's just continuous. So why,
And
right?
yeah, why did that change? What was what
Yeah.
what occurred where that changed? And I mean, the one you showed us was looked almost more like a sailfish. So
Yeah.
at what point did the marlins start having just a short dorsal? Um,
Yeah.
and
Yeah.
whatever that is. So yeah, that's interesting.
Yeah. There's sort of this law, or I don't know what to say, like the statement in evolutionary biology, which is that ontogeny, so the development of fishes, what is it? Ontogeny follows ancestry. I don't remember, but that if you take an early stage of an animal, that early stage of that animal looks more like the ancestral animal than what the adult looks more like. So I mean, this is in that, like, I really don't know if the fishes that I'm looking at, if they're fully grown or not. And I so far I have not read if anyone has studied what stage of development they would be on. But who knows? Maybe these what we're getting, these smaller specimens, are just juveniles too, or maybe they're fully grown adults that are just tiny,
Yeah,
like sort of the juveniles that we see nowadays.
yeah, that's interesting. A lot of mysteries there, I guess.
Yeah, yeah. There's a lot there. Work. What is it? Job security.
Yeah, yeah. There's always questions. I mean, some of them
Yeah.
pretty hard to answer. So
Yeah, don't tell that to my advisor though, because he'll be like, "Well, you're not going to be a PhD
yeah,
student forever."
right,
Yeah,
right. So I guess the next thing I'm curious about is the current stuff that you work on,
yeah.
and like and the small ones, the really small ones, the larger ones, everything. Like how do you get those
Okay.
specimens, and how do you go through it, and what's the whole process there?
Yeah. So it has been one of the most enjoyable parts of my PhD work, is being able to go to fishing tournaments and sort of getting specimens, actual specimens. As I told you earlier, I had never seen a billfish before, like in the flesh. And the first time that another professor from VIMS, she does a lot of work with genetics and like population dynamics of billfishes and all that. And she was like, Miguel, well, you got to come with me to the Mid Atlantic to get some samples. I'm like, heck yeah, that sounds good, Jan. So we went to Ocean City, Maryland, and the first thing, the first fish that they brought up was a blue marlin. I remember that, and it was not even that big of a blue marlin. It was like 400 plus pounds, but I was just like mesmerized, like what the heck? There's no way this exists. It's just so cool. And some of the anglers, or the angler from that caught that fish, was super generous, and The him and his crew donated the head with the bill and everything for my research. So that was amazing. So like with the help of the Billfish Foundation and the Mid Atlantic, it has been great. Also, I don't know, I haven't mentioned it yet, but I got a scholarship last year from the Billfish Foundation, which has been just like really great, like really helpful for my research. Especially getting imaging equipment for like the photos that I showed you. I am a tinker, and during the pandemic, I went a little bit crazy, and I ended up buying like 30 microscopes, and I put together like a dissecting microscope to take good photos with. And the only thing that was missing was to get a nice camera with it. So I like that they helped me a lot to get those nice photos. But apart from that, getting in touch with the Mid Atlantic and everyone there, like the Billfish Foundation, has been just great at it. But yeah, so we get the specimens from there, or the ones that I've prepared from there. And yeah, we rely on the generosity of anglers. And so whenever I get a nice whole specimen, like let's say a white marlin that we get donated, I usually like to use it for more than one thing. So yeah, my research, like it's a great use, but if I can use it for more, that's great. So I usually end up doing like life, not like dissections on in situ of the animal to show people sort of like the anatomy of them, and people really enjoy those. And it allows me, dissecting that allows me to do the first stage on the preparation of these animals for my research, which is this, and show you, is just getting rid of most of the meat. So here we have a fish that I've taken some of the meat out. You can see some of the features. Did you know that they have really? Oh, I didn't know that they had such cool swimming bladders before, and they're just the most amazing thing. So they're like these little bubbles are
Yeah.
what their swimming bladder is, right? They're just so cool, and like it goes all the way like to the back of the fish. It's just like such an amazing thing.
It's crazy how far it
Um,
goes back into the fish. I didn't realize they went that far back.
Yeah, yeah,
Yeah.
yeah. It goes really far back. And I remember the first time dissecting it, and I was there with Jan. I'm like, Jan, what are these bubbles? And she was like, that's a swimming bladder. I'm like, I had no idea. And they look cool because you open it up and you see a bubble that's sort of metallic-y looking. It has like a metallic sheen to it. And it's just all those compartments, like all the way to the back. So first step is just taking most of the meat out of the specimen. And then what I do is I let it dry. So I let
Oh,
them
wow.
dry with most of the meat taken out until it's just like fish jerky.
Yeah.
um, and the way you do it so that it doesn't rot or anything is we just put it in a fume hood that has good airflow, so it dries before getting mold on
Yeah.
it,
Kind of
right?
like jerky.
Yeah,
Yeah,
yeah, yeah, yeah,
so jerky.
yeah. Just like that. And so this is after taking it out of the fume hood, and I just put all the parts here. We have a big a blue marlin head. Oh, this was the one that I was telling you was the 400. I think it was like
Right.
420 pounder. And then from on the last tournament last year, they actually donated a whole blue marlin, and this is the caudal fin of that one. I don't remember exactly how much it weighed. And you can see I have a tuna, a big eye tuna back here,
Right.
and another assortment of a white marlin and us and spearfish. So they go, yeah, after taking it out the fume hood and making it into jerky. We put them in this enclosure, which I was a new enclosure that I had to build it last year because the enclosures that we had before were not big enough for the specimens, so they were little. And the way that we clean them up is using these little things, which are dermested beetles. So I don't know if you have ever seen how they prepare taxidermy animals like deer and other stuff. They usually use beetles, and what the beetles do is they eat all that jerky, and they end up leaving a nice clean bone or bones for us to use. And so that's why I needed an enclosure, because if not, they escape, and it's not good because they're like my office is in that lab, so if I have a
With the
ton of
Beatles, yeah.
beetles escaping, that would not be good. But they're really cool. And yeah, so, and here's a photo of that caudal fin with some beetles on it. And these are the adult beetles,
Mhm.
and these are the larvae or nymphs, and they just eat all that. They're the best pets to have
Yeah.
because you only have to spray them like once a week, and if you forget, they'll be fine for several weeks at a time. You
Yeah.
spray them with water, and then they just stay there. They're very low maintenance. Um, yeah, but after they're done with cleaning those bones, I take them out and then just brush as many beetles as I can back into the enclosure. There's a ton of beetle sawdust, which is a mixture of beetle poop and just molts. So when they go from the larval stage to the adult beetles, they molt, and they molt several times between that. And so you clean them all up, and then you put them in a freezer for a few weeks because there could still be some living ones, and you
Right.
really don't want any living beetles to stay in your nice specimens for a long time because they can start making damage to like other parts of the specimen.
Right.
And then after that, if the bones are oily, like what you see in billfish skulls, they're very very oily. What you do is you soak them in ammonia, and that gets rid of all that oil. And then after that, they're ready to either for me to study them or for them to go into the fish collection, where they'll be stored in perpetuity. Sort of.
Yeah.
Yeah.
Well,
So that's sort of the process
yeah.
of
Yeah.
getting them.
That's pretty
Yeah.
cool.
Yeah.
Yeah.
I didn't realize that. I was curious what you did. If you just put them in a cooler and brought them back, and then just started going to work on them, or what? I mean,
So I'm a little
Yeah.
dramatic, Chris.
yeah.
Yeah, and so this is the preparation that I'm using to look at the teeth, um, very at all the teeth, like dried specimens. But you also can, if you have a nice specimen that you want to keep as a whole with all the muscle and everything, then you inject it with formalin, put it in formalin for several months, and then you wash it with water to get rid of all that formalin and store it permanently in ethanol. So that's the other way of doing it. But I love working with the beetles, so like that's just a good excuse, and they're not as stinky as you
No.
might think. Like it's not bad. It's not bad at all. People always look at them and are like, ah. I'm like, yeah, they're not bad. They're not so bad. I
That's
know I
interesting.
know of some colleagues that they have worked with beetles for a long, long time, and they start getting allergic to them. That
Right.
has not happened to me yet, or I already have sinus problems, and who cares? Yeah.
Right. Oh, that's interesting, man. So what the really small ones you get, is it a similar process that you would go on, or like
So
drying them out and everything, or how do you keep those?
So for the really small ones, so the larval billfishes, those are all stored either in glycerin after
Uh-huh.
um, they're prepared through clearing staining, or they're stored under ethanol.
Okay.
These I have more photos of juvenile billfishes here, which once again the best looking fishes ever. They usually get stored in under
Oh,
ethanol,
cool.
so they get fixed in formalin and then stored in ethanol. So yeah, you can see that. Like this was that specimen that I showed you a photo of. But yeah, I have some different stages here.
So
Um,
at that age, they all have that kind of sail, right? Like even the blue marlin and stuff, or at what?
so this was something that I found out like literally last week. Is that blue marlins look very different
Oh, they at
than
that age, at that size, they do look different.
they look very very different.
Oh wow.
And let me get a photo of one. Um, um, yeah, they look very very different. Um, and here's one from the internet. Um, and I know I had found some better ones, but yeah, but you can see that it does have the really big fin on the back, but its bill is not as developed, right?
Yeah.
So they're just
Yeah,
weird.
Bill's nowhere near as developed. Wow.
Yeah, yeah. So and this is probably a sailfish, but yeah, yeah. Some of those stages they just look very different. And this is something I found last week. I had no idea. I had
Yeah.
no idea that there would be that different. And now for sure I need to get some specimens of juvenile blue marlins because they'll be really neat to see the how the teeth are on that stage if they don't have if the development of their bill is slower, you know.
Yeah.
Um, so so yeah, that's something else to add.
Yeah. Oh,
Yeah,
that's interesting.
but if you're ever night lighting and you get a blue marlin or a little sailfish or anything, um, of course there's some permitting involved or anything for us to take it into the collection or for me to use it as part of my work, but I really need those, like some of those specimens.
Right.
For
Right.
sure.
Yeah. That's good to put
Yeah.
out there. Just let people know if they come across them very often or catch them in nets or something like that, that it would help.
Yeah, and I was telling Chris earlier that I had some leads on mahi, but like from some angler, an angler that was telling me that when he catches mahi, sometimes they regurgitate juvenile billfishes. And I'm like, no way. So if you know if you see mahi and decide to look into her stomachs, keep an eye out for juvenile billfishes. And actually, after that, my committee member Jan, she went to a tournament and she sends me a photo of two swordfish that were like this big that she had actually extracted from a mahi stomach. So they're definitely eating juvenile billfishes, but it would be really cool to get some of those specimens. And I
Gotcha.
like I don't care if they're a little bit digested. They don't need to be perfect.
Yeah. Yeah.
Yeah, yeah, but that's cool. So yeah,
Dude, now I'm curious. I got 3 mice in my fish box right now. Like
yeah. Nick,
There you
you
go.
gotta go and get
I'm
them.
curious now. Like
Bring them in the stateroom there and just bust them out there.
Yeah,
Start.
Yeah,
yeah, I'll probably right on the new duvet. I'm sure the owner will be real.
Have a look, though, man. I mean, I would think if that's where it's going to happen,
like
it's gonna
now that
happen
you said
right in the
that, like I'm
Bahamas.
100% gonna cut the stomach of the mite. Like I normally don't, but now I'm going to. Like
Yeah.
And if you find some, please let me know. Yeah, just put them in a bag, freeze them. And
well, I'll let you know because the mites over here like a bunch. Like we reel baits away from. So if we do find them, I'll start like kind of like actually.
oof, yeah, that'll be awesome. If there's stomach contents, I think that the permitting would be easier. Uh, I'm not sure, but I think it might.
I think you'd be fine. It's also kind of funny to me that the mahis eat it, the blue marlin grow up. They're like, oh yeah, joke's on you boys.
Exactly.
Like, oh, yeah,
Change this real quick.
You and my brother Bob watch this.
yeah, for sure. Yeah, payback.
Real quick.
Oh, yeah, no worries.
Yeah.
Miguel, when you were talking about the swim bladders, have you done or at least spoken with people much about the functioning of them or anything about them or anything, the complexity of it and kind of what they're doing?
No, I have not talked to anyone.
Okay.
I think that I heard that the swim bladders in billfishes do not change size to change buoyancy, but I'm not sure. I think that I heard about that, but like now you put that in my mind. Now I have to find out. I have to ask around if anyone has done some work on swim bladders because they're definitely remarkable, and the fact that they go all the way to like that far back on the fish probably has to do more than like vertical like where they're vertically, right? Probably more of what they do this way, right?
Yeah. Keep them,
so yeah,
their orientation
stable in that axis.
I caught some before that have come up and just turned upside down, and we're still pretty alive. And
really?
We're kind of swimming and kicking. Yeah. And man, my trying to save those things when they do that, it's really hard, man. I've tried to get them back down deep. I've tried all kinds of things, and they always seem to just come and float back up, tail up, and then kind of come like this. But they're still alive. So I'm thinking what's happening now, especially seeing where the swim bladders are fully located. It's like they must be filling up with air, and they can't. They break the swim bladder in some way, and I mean that's interesting because that's exactly where they're floating from. It's like that back section all the way back.
Oh, really?
Tail up.
Yeah, so that's interesting.
Yeah, that's very cool. Yeah, that's very interesting.
That might be something, like because when guys are bottom fishing and they bring up fish too fast, right? They like inflate, and then there's like a little tool you can get, like to get them down, or even like a weight to get them down there to get the pressurization. But I wonder if that's something that could be done with marlin, where you could just use that tool, but like find where the air bladder is instead of, because if you do it where you on those bottom fish, you just end up puncturing their stomach. It doesn't work too well.
Well, I'm wondering, Chris, if whenever you have a problem with a marlin that has had some bladder issues, maybe that would be a specimen worth sacrificing and see what kind of damage it suffered. Because
I'm sure it happens every once in a while. I mean, we had one like Nick said, it was with Nick actually. We had one and we ended up we tried to get it back to life. It just we wouldn't. We tried for a while. We ended up just pulling it in the boat. So I mean, that's something where having someone, a scientist or someone here in Hawaii, because it's kind of hard to get a 400-pound or 500-pound blue from here to you.
I can always come to Hawaii. I don't mind.
I mean, I'm surprised they don't have some of you guys come out for like the tournament series in the summer, because there's quite a lot of big billfish that get killed then. It's all kill tournaments, so it's all fish over 400 pounds are getting killed, and that runs pretty consistently through June, July, and in August or something. But from what I know, most of that meat just gets donated or like smoked or something like that. I don't think there's a lot of like research being done on it, but I could be wrong. Jody may be donating some of it to some people, but I don't think so. Any fish I've ever killed, it just gets eaten.
Yeah,
But,
I think that to tournament that I go to, they also donate most of the meat. Yeah.
yeah.
But yeah,
That
it
was,
would be
to learn a bit about those swim ladders. And
yeah.
I did an article a long time ago about billfish hearing. I did 2 articles in Marlin. I did one on billfish hearing and then one on like boats and different things on whether they raise more certain sounds or certain boats. So I had contacted a bunch of different people that work in that field and scientists and stuff. And it was a while back, but I believe that their how they sense sound is linked in with that swim bladder as well. So like there's vibration that cavity is like creates a vibration or something that then travels into how they hear. And that's kind of how they like could feel a boat coming when they're 67, 50, 60 fathoms down, and they can't see it obviously. And I don't think they really can like hear it like we could hear it audibly. So I think that there's a vibration that they sort of feel in that swim bladder that then sparks their interest and they go up and check it out. So I think there's a lot more to those swim bladders than just being buoyancy or whatever, which is be interesting to really kind of dive into it. I like I said, it was a while ago that I did that, so I'd have to go back and kind of read what I wrote down and who I talked to. But yeah, it's a cool aspect. I mean, I guess you could get lost in. There's so many interesting things that these things have,
No, but I know that, and that swim bladder is definitely remarkable. I think it'll be worth looking more into it. I'll ask around and see what people have done about it, because it just sounds like a cool thing. Yeah. Especially if it is related to hearing. It has big implications for angling, right?
Yeah. I mean, that's what I was trying to figure out and see how they hear, what they hear, and what's going on with all that. And what was the other thing that I found interesting was the lateral lines. I had always had the idea that lateral lines, they like use the lateral lines to find prey and stuff. But the scientists that I'd spoke to said that it's more for spatial orientation. So like schooling behavior or like being aware of things around them. So they may use it when feeding, but it's only those last few seconds. So like for us, like right before they strike a lure, the lateral lines playing a part. Whereas in my mind, I'd always thought, oh, they pick up the vibration from the lateral line way down away and they hear us and then they come in where it's their otoliths and whatever they're doing with the swim bladder and the vibration. And then as they getting close to feed, then they're using their lateral line. So
that's just amazing.
Cool stuff.
Yeah. I wonder how much, I mean, there's so many different aspects they're using to feed, like how much is actually visual on top of that, and how optically, because we put so much, as anglers, we put so much attention into like color and rigging and like, okay, can they see the hook? Can they see the leader? Can they do this? Can they do that? And how much of that plays a part in actually the bite you get out of the fish, as opposed to really like what they're feeling and sensing, and they're already committing and they're coming in, and whether the optical part is really only the very last little bit or not. And from what I know, I couldn't get like a whole lot of information on it. It's like something that hasn't been fully like looked into, but we have our theories, I guess.
Yeah, of course. Do you see a big difference in performance related to like the color, or the like patterns that you put in lures?
Personally, for me, not like a big enough one that I care for marlin. But tuna are different, and I mean, I'm sure if you talk to anyone that's done work on the eyes, they can tell you too. Like the tuna eyes are probably a lot more sophisticated and more attention to that kind of stuff. But with the marlin, I think sometimes it matters, but I don't know, man. I think that they're already there. That's reaction and movement and feeding, but everyone will tell you something different. I know, there's some amazing captains out there that are going to tell you that no, color is like huge. It's very important. You got to have it, but it's it's just a conundrum. Like, and if you go down that road too, or it's like, well, it's a lot of guys will say, well, you just got to match the hatch, right? So like, dress your lures up to look exactly like the bait that's out there. But here's the funny thing about that theory is like, so, all these fish have evolved to survive. So the colors of these fish is evolving so that they can camouflage, so that they can hide from fish, so that they can, it's harder for fish to feed on them, right? Like the ultraviolet stripes that come out in some fish, or the mackerels, the weird patterns on their skin is to, kind of confuse a fish or whatever, and all these things. So then if we're dressing our lures up just like these fish, well, then we're kind of like screwing ourselves in some ways, because we're camouflaging we're using the evolutionary tactics of this animal to hide from its prey, to then hide our lures from this prey. So wouldn't it make more sense to put something out there that's just like dog balls, and that thing comes up and it's like, oh yeah, that's an easy meal. That's an easy meal right there, dude. Yeah, look at that thing. I've never seen a pink fish before. I'm eating that, I don't know. I mean, you can go
it's a good point. I find like people say match the hatch, and I've definitely said it. I learned from Chris, so I do more like I try to think of my spread how Chris taught me how to think of the spread. The only thing I'll add, like if we're like in an area where I'm seeing like Dorados everywhere, I'm gonna slide a green lure out. I'm not gonna change my whole spread, but I'm gonna slide a green out, and generally speaking, I'm probably gonna get bit on that. But like I won't like change everything for that. Like, I'm still gonna have the kind of the base spread of what Chris taught me, what I kind of want to run. But like if you see a bunch of one thing, maybe slide one that looks out like that, like similar to that kind of thing. That's kind of how I I try not to change my whole spread based on what I'm not going to change all the. I'm not doing that.
Yeah, that's very interesting. That's so cool. So all the biases that go into selecting what lure, what are you thinking that fish are gonna
you can really stump yourself if you think into it too much. And then there's the guys that do like if dark days, dark colors, or bright days, colors, and all this. I don't know. I mean, I don't know. If you ever video lures underwater from 10 feet away, it just looks like a bubble with a flash. I mean, with barely any color. It's just very difficult to see that color until they're right there. So they're probably just seeing flashes of color really in there. But hey, it's whatever. At the end of the day, I think the important thing is like the captain and the crew to feel comfortable. So if you got some lures out there that are in colors that you're just like, I don't know about this, like it's all fluorescent yellow, I don't know if that's going to get bit or something like that. Well, that's not a good feeling to have. Whereas if you got stuff out there, you're like, oh yeah, this stuff looks good. They're going to eat this. And that's probably a better mindset to have, and just to have out there and do it. And the contrasting colors is probably important as well, I would think. And depending on the lures too, like we have bullets that we pull, and they don't really make a bubble. They don't make much bubble. They kind of just wiggle, and every once in a while will pop. So those are going to be you're going to see the color a lot more on that than like a big plowing lure that has this like huge bubble of water around it, of air around it, and white water and all this kind of stuff. So but
how deep do you usually because I have nothing, I don't know anything about billfish angling, and I should get more acquainted with it.
You need to just get on some boats, go out. And if you're,
it has not happened yet, but I gotta convince someone to take me on their boat and just explain me how it works. But usually, how deep do these lures, like do you deploy these lures, or how deep do they go, or like does it vary? Like, is there?
They're not very deep. At least what we're doing. There's some guys that will rig like baits on downriggers and do different things differently. But like for pretty much marlin, blue marlin, trolling, lure trolling, the majority of our lures are not going that deep. You might have one one that goes down and gets down there and digs, maybe goes 6 feet, 7 feet down there, maybe a little more, maybe even 10 if it's like a really good plunge. But most of them don't go that deep. It's a lot of surface activity. So our bites are all surface bites, which makes it exciting too, because you think about a big marlin coming in and eating something, and they don't do it very delicately. Sometimes, usually it's like a plane crash, and it's right there and it happens in front of you and it makes it exciting. So, but yeah, most of them are right there sort of at the top. So, they kind of come up and see that bait sort of stuck between the surface and that area, and it, doesn't have anywhere to escape. So they come up and eat it.
And even Miguel, we're fishing like over here. We're not really fishing that many lures. We're fishing like dead bait, and we're running all surface stuff.
Oh, surface. Yeah. Okay. Oh,
yeah. Like saying, we had the other day, Chris. We actually pitched a double. Well, we had a double. We call it double blue. And we had one. We're running like our dredge. We're running our dredge and like a dink on top of the dredge. Then our first teaser, and dude, we're just sitting there looking at the dredge or the teaser, and this freaking blue marlin came up like a missile and just ate the dink, like just ate the flat line right there in front of it, just out of nowhere, full roll.
That's cool.
Like, don't know where that came from. We don't have a sonar, but it was like, don't know where that came from. Like, all right, I'm feeding it.
Yeah. Oh my gosh. Yeah. Last year, during the tournament and everything, we got another blue marlin donated to us, and we did a stomach content thing, and we find a full teaser inside the stomach.
You did? No way.
Yeah. Yeah. And then I remember it was like a squid-looking red thing. My colleague Quinn, she has it by her desk.
That's awesome.
And I remember just asking, like, did anyone lose this? Because it was not. I don't think it was the angler that caught it. It was, someone else. Yeah, it was really cool. I have another question for you, guys. What kind of line do you use? Because like these things have like the force involved in catching them has to be amazing, right? Like.
Yeah. I mean, everyone uses something differently. Here we're using mostly 130, maybe 80.
And that's that night.
And then the leaders everywhere's different. What they're doing where Nick's at right now is probably all 20 and 30 pound. I would think.
We use a 30-pound main line to an 80-pound line leader, but we don't have a big fish. We have a big fish like 80.
You have an 80 or a 50 pitch or what?
We have a 50 and an 80 pitch. We just kind of go back and forth between days, and we have that on like 400-pound leader.
What are you pitching on your 80? A mackerel? Your big pitch?
Horse balladoo with juggerhead.
Oh, really? Even on the big pitch? Oh, wow. Interesting.
Well, we just get more day. I mean, you could do a mackerel. We just get more days out of them, dude. You can like those balladoo's.
No, mackerels are super expensive too, and where you're at, you probably only see.
Well, I'll text you later. I found some insanely good mackerels for you.
Oh yeah, okay, yeah. Message me. I need.
I'll text you later. But yeah, we're mostly using 50. But yeah, Miguel, we're using a different leader around those. Actually, now that you've told us it's teeth, I just thought it was a rough bill. We're using a thicker piece of leader right around that.
Okay. Yeah.
But main line, Anyway,
yeah, because those teeth will chafe your leader to the point of like breaking your leader off, even heavy leaders. So like here in Kona, where we get really, really big fish, we're using 530-pound leader.
Oh my gosh.
7 feet of that, and then our main line's like 130. But even beyond that, the like the hook rig itself, which is basically what's going to be kind of in their main part of the face, like in their jaw. Okay. We use 800 or 900-pound cable, like arrow.
Oh wow, yeah.
Cable. Yeah. Because it's crazy that I've seen it can happen. The really, really big ones the way their jaw hinges, if that line gets just the line, not so much the cable, but the cable they can't break really. But if the line gets in that little like jaw hinge, the really big ones have so much force they can like smash that line, or they very much crush it enough where it loses strength and you end up popping it off.
No way.
And they'll like tweak hooks and stuff in the corner of their jaw. They'll be able to bend metal and stuff. I mean, it's crazy.
Bolt cutter.
I almost lost I wouldn't have lost my finger, but I cut it very badly in a billfish in his mouth.
Oh, really?
I was trying to get a hook out, and my finger was right in the corner of his jaw, and he hinged down. I was like, and I had the gnarliest slash in my finger from a billfish. So those really big ones can tweak it. And I mean, I've never fished the Great Barrier Reef in Australia, but a lot of those black marlin captains and stuff will talk about those fish being able to do the blacks being able to do some pretty extraordinary things with their jaw hinge and with their mouth, and just the pressure of, that they can, because if you think about it, they can crush tuna skulls and just
those big ones. It's got to be, I don't know what it is. It's got to be something unreal.
So my colleague Laura, she actually did a study of the bite force on them, but it was like a model, like she used computer modeling and all that to estimate it, but I don't remember how much that was, because like if it is bending hooks, like and the hooks you use are thick, right?
More so that's happening on a smaller hook, when someone hooks a big fish with a smaller hook, but even like an 8-0 or something, they can they can tweak it.
Too, like it's just over. They may not pinch it immediately on one time, but if it just sits there and they just keep
doing and moving, and it's gonna start thing. But they're powerful, man. The big ones are very, very powerful.
That's so cool.
Impressive, man.
Yeah, Chris, if you could send me, you said that you had some lures that were scratched by the teeth.
Yeah.
If you could send me photos of that would be awesome.
Yeah. All our lures are scuffed up. I mean,
you got a lot of them.
Yeah, I mean, shit, I got one laying right here.
Oh yeah. Oh my gosh. Yeah, that's so cool. Heck yeah, that's awesome.
They just look like little kind of like rash marks, but yeah, I mean, we get, they definitely scrape them up.
They do. They see down there and up there.
They're like little teeth marks,
Yep.
Now that we've had this episode, like that actually makes even more sense to me.
Yeah. Those are really, really cool. But yeah, if you could send me some photos of those, I think that would be a really cool figure, and I'll credit you for sure. To put in whatever I end up publishing.
Right.
Yeah. That'll be awesome.
Cool. Well, I'm pretty much out of questions. Is there anything you want to go over, Nick, before we?
No, I mean, I think he just dropped a knowledge session on us.
Yeah.
I mean, the biggest thing, I think I asked him. I was so excited. I think I just asked him in the moment. I probably hijacked it a couple. I think he hit everything.
Yeah. And what I've learned from you guys is awesome too. No, this is so cool. I gotta keep learning more about the art of angling. Billfish.
Yeah, you have to get out and fish for sure, man. Check it out and see these things and what they're capable of.
From someone who studied them and fishes for them, it's seeing them in flesh. Miguel will never get on.
Yeah. And some people have told me that the coloration when you're catching them, the blue, and they're like, what you see here is nothing. I'm like, no way. And like, I would love to see that. Yeah, that's awesome. And I'll definitely ask around what if I can go to Hawaii for maybe one of the tournaments or something. That would be amazing. Yeah, maybe I don't know if my advisor would like it or not. Like, you need how much money? I'm like, yes.
Yeah, well, if you get over here, we'll take you fishing, man, for sure. So if you get over,
Awesome.
let us know.
Uh, that would be great. Yeah, thank you so much. No, I really enjoy that. Yeah, and if you happen to know of anyone that ends up catching like a black marlin or a good sized swordfish that I can get ahead with the bill intact, that would be so helpful.
Yeah, I'll ask around. I mean, do you need the whole head or just the bill?
I would need the whole head. Yeah.
Okay.
Yeah.
All the way back to about where the gill plate ends, kind of deal.
Yeah. Just about there. That's how we usually, for the bigger specimens, that's how I usually cut them. Just right there.
Yeah. If I hear anything, I'll let you know. But like I said, Cabo is probably a good option.
Got it. Cabo. Yeah.
With a few of those guys, because they kill everything, and
okay.
Quite a lot of marlin. Everybody complains about Hawaii. Like, come on, we release most of our
fish. Yeah, they talk about us.
Like, Cabo, they whack everything. They're whacking like
200
marlin and 800 pounds.
You could probably talk to Peter too, Miguel, about swordfish. I mean, half the guys,
guaranteed.
deep drop. I mean, I'll talk to my captain on this boat. He's from Palm Beach. Like, he's got 10 friends, commercial sword guys. Like, there's no way.
Heck yeah.
Yeah.
All right. Yeah. No,
so
that'll be
that are sword fishermen back in my being back, but like he's got to.
Okay, so, I'll definitely talk to Peter about that. That'll be so cool. Yeah, and so what I was telling you that I usually dissect fishes and it's during our open houses. I started doing this like the first year that I was here. That was at school, and now I don't have a cool blue marlin head for this year, so I really need to get a cool swordfish head to dissect. And just like kids are the best, they do not care about getting their hands dirty. They come up to it, I'm like, can I touch this? I'm like, yeah, sure. Do you want to use some gloves? And they're like, no. I'm like, okay, but the sink is down there afterwards. Yeah,
that's cool. Yeah, well, I mean, you're in Florida, perfect place to get some swordfish heads. I'm sure you just put that.
Yeah, I mean,
ask Nick said. Ask Peter. He's probably got a list of people who could.
Yeah, I mean, he's got people on that pay in. They go swordfish and help those guys.
Yeah,
perfect. All right, I'll ask Peter then. Yeah. Thank you so much, guys. No,
thank you.
That was so cool.
Yeah, no, thank you, man.
Appreciate it, and we'll be in touch, man. I'll email you through some pictures and stuff.
Awesome.
And then when we get this thing all the recording and everything too, I can send it to you if you want to pull any clips or anything off it, you can or whatever. But
awesome. Thank you so much.
Well, thanks, man. I appreciate the time, and we'll have to do this again sometime down the road and see where what you just learn as you go.
You just let me know. Yeah, maybe we get into some of the swim bladder mysteries.
Yes, please let me know.
Cool.
If you learn more about those. That would be
if you learn more.
Yeah.
And Nick, if you find any partially digested billfishes inside your mine,
I'm legitimately gonna keep it. I can put it in the freezer too. I'll legitimately keep a lookout because we're kind of like putting the hurting on the mahis right now. Dude, they're everywhere.
There you go.
Okay. Yeah, that'll be cool. And so another thing that, like, if you do find any, maybe just like put a note on like either how long or how much the fish weigh, because it'll be nice to know at what size they're feeding on billfish, right? Like that'll be like another side project or something that someone else can take over. But it'll be an interesting data set
when they're eating them, right?
Yeah, especially for people that do conservation and ecology and all that. Yeah, I am not a statistician, so that's like I count bones. I'm like, there's 2 bones there, there's 3 bones there, but nothing else. Yeah, but cool, awesome.
All right. Well, thanks, guys. Nick, good luck fishing tomorrow, man. Give me a call. Give me a shout when you get home or when you get some time.
Yeah, I'm get time. We're actually gonna take off tomorrow and go Saturday, but I'll give you a shout later next week. We have some time.
Cool. All right.
Miguel, it's nice meeting you, brother.
Nice meeting, Nick.
Yeah. Thank you
Alrighty.
guys. I'm going to hit the